These worms are literally eating their way out of this plastic bag. But not only can they eat it, it's actually good for the planet. So if they can biodegrade plastic, could they be the answer to our planet's massive plastic problem?
Biology has found a way to some extent to deal with this.
Because the latest science on this is mind-blowing. Not only might we make plastic biodegradable, we might even one day be eating vanilla ice cream from recycled plastic and E. coli.
Yes, I mean, it's chemically identical.
Yeah, so we'll get back to that in a minute. Let's start with the worms. These little creatures are waxworms. Dr. Federica Bertocchini is a molecular biologist, and she first witnessed this phenomenon when she chucked a bunch of waxworms in a plastic bag as a hobby side project, because, well, who doesn't?
I bumped into the waxworm accidentally because at that time I was a beekeeper, because they live in the beehives. They are considered plagues by beekeepers. So I started cleaning my beehive, putting the worms in a plastic bag, and within a short time I realized that they were producing holes.
The plastic started to degrade almost as soon as it touched the worms' mouths.
So we thought, okay, maybe something coming out of the mouth. So we start collecting this liquid coming out. It's called saliva, but it's the liquid coming out of the mouth. So in this saliva, we found two enzymes that can reproduce the effect of the saliva, meaning oxidizing polyethylene. And it takes a few hours at room temperature in a watery solution.
And the amazing thing is that the worms can even digest the plastic, breaking it down into something useful for the worm.
The worm itself, when it eats the plastic and starts breaking it down, its guts react almost as if it was eating normal food. So that means that there's something happening with the physiology of the animal that extracts something out of this plastic biodegradation, and it just continues as if it were a normal diet.
That's Dr. Chris Lemoine, who, inspired by Federica's findings, also began looking into these worms.
We found that the plastic allowed them to still retain all their fat and presumably continue with their life cycle.
Basically, these worms are fattening themselves up by whatever means necessary before they turn into moths, by which point they don't eat again, only reproduce.
I always call them bags of gonads that can fly, because that's all they do.
So there's a race underway to figure out just how this mechanism works.
That's the $1,000,000 or $1 trillion question, because once we figure that out, that's $1 trillion worth of plastic we can degrade.
Because as cool as the waxworms are, this is really about the specific combination of bacteria and enzymes that can break down plastic, something that's exceptionally rare in nature. So why is it so rare? Why is plastic so hard to break down? Well, in nature, most things decompose because bacteria breaks down the chemical bonds that hold a substance together. These enzymes and bacteria have evolved over millennia to break down whatever it finds in front of it. Then plastic comes along.
Here's a scene that has long since ceased causing any surprise. Dishes that bounce when they drop to the floor.
Nowadays, it gets a bad rep, but it's a total game changer for humanity. But nature's never experienced it before. Plastics are made up of long chains of polymers with very strong bonds, and one of the keys to breaking these bonds is through oxidation. That's what the worms appear to be doing with their saliva, introducing oxygen molecules to the plastic.
And this is something that's achieved in environment through light, for example, high temperature. And this is the bottleneck. It takes a while because the environment has its own timing. So the worms, what they do is just introduce molecule of oxygen. So in a few hours instead of months or years or whatever. So this is it's a way to overcome the bottleneck of this reaction.
So what's next? Unleash the worms? No, that would be a terrible idea. Remember this?
They are considered plagues by beekeepers.
But even sticking just to plastic, the process is still way too slow to realistically solve our plastic crisis anytime soon. So stand down, waxworm armada. The real stars of this though are the enzymes. If the researchers can identify them and scale them up, there's a chance that in the future this could be one of the solutions.
It will take a lot of cash.
But now scientists are looking for similar enzymes in all sorts of other places.
Superworms too. So anything that has a worm in it, it seems to be prone to eat plastic.
In fact, over 30,000 enzymes have been identified capable of digesting 10 different types of plastics. One bacteria found in cow stomachs can be used to digest polyester. But the one that's getting everyone really excited is a bacteria called Ideonella sakaiensis, and especially its enzyme, PETase.
Plastics waste in general, but more specifically PT, has infiltrated our environment. And Biology has found a way to some extent to deal with this over time. There was a discovery outside of Japan where they had found that microbes began to colonize on parts of a water bottle. Cells were actually living and maybe even surviving off the carbon within that plastic. We can take this enzyme out of the cell and we can begin to engineer that enzyme even further to be able to have better activity directly on plastic waste.
PET plastic that would take centuries to break down in nature, PETes can break down in a matter of days. But it doesn't solve our plastic problem. Not yet, anyway. To have any real impact at scale, we need to turbocharge how quickly it works. And that's exactly what Hal's team has done. And they named this really fast version of PETes, fast PETes. They did this using AI, essentially by using a vast database of all known enzymes in the natural world and then running simulations about which combinations and mutations would speed up the process, kind of like a form of computerized accelerated evolution.
Machine learning approach really it's rapid evolution to some extent on there, but at the same time, it's guided by observation. We saw that this enzyme was not very stable overall, and used a machine learning type of approach to figure out which point mutations would make this enzyme more stable, and found a couple of mutations that really both increased the stability significantly, but then also gave rise to a significant increase in the activity of this enzyme on plastic.
This cutting-edge technology opens up a whole new frontier of scientific possibility, and the team aren't done yet.
Thinking about trying to clean up plastic that's actually in the environment. Those applications don't have the benefit of being able to control the temperature and pH very well. So having an enzyme that ultimately is flexible enough to work in a variety of different conditions is extremely valuable.